In this video, we will discuss about roles of chaperones in N-Glycosylation. So first we will understand uh, what is the motivation to understand the roles of chaperone in N-Glycosylation. So the story begins with recombinant glycoprotein production. Most of the therapeutic recombinant glycoproteins that are produced do not use any man-made manufacturing machine. However, they used already existent cellular machinery such as mammalian cells, insect cells, yeast cells, prokaryotic cells to produce their final recombinant protein. So things start with they have their target gene that can produce their therapeutic protein. They uh, convert them or construct an expression plasmid of this target gene and then transfect it into the uh, one of the one of the expression system that can produce the recombinant protein using their cellular expression machinery. So in this case, in the case of different cellular expression, different cell expressions or different types of cells available for expressions, each one has different advantages and disadvantages. So as some of the advantages and disadvantages are mentioned here, such as this bacteria has very limited post-translation modification that might lead to immunogenic reaction. The yeast is the therapeutic glycoprotein produced in yeast has uh, hypermannose glycosylation. So this will produce high mannose uh, proteins, and our human beings are not human beings post translation modification does not have the high glycosylation structure, so it will produce a immunogenic reaction against these proteins that have high mannose structures or glycans. Plant cells has a risk of this glycosylation. So most of this glycosylation production platform has the problem with the post-translation modification, as the post-translation modifications are not the profile of post-translation modification are difficult to match with human glycosylation profile. So there is a push to use this uh, cell lines derived from humans for the production of therapeutic glycoproteins, and other things are also need to be taken care of, as it is highlighted in the red circle, is that the best uh, or the better glycosylation production platform will have human glycosylation profile, is recombinant protein yields will be higher, genetic modification, it is easy to uh, modify the genes so that we can produce the gene of interest easily in this within this platform. It should easy, or we can say the cell lines should be easy to transfect, and there is low risk of contamination and minimal, and it does not require very expensive raw materials, or we can say minimal or inexpensive media, and cell growth should be high so that uh, amount of final product or the proteins that is produced from the same amount of cells will be higher. So these are the criteria that needs to be fulfilled in the future glycoprotein production platform. Present scenario, one of the mammalian cells, that is the CHO cells, can produce 70% of the therapeutic recombinant proteins. However, this also has problems related to post-translation modifications. The future possibility is that use human cell lines for therapeutic proteins, that will reduce the risk for immunoreaction. And in order to improve the problem with the human cell lines is that the cell growth is very low and the media requirement is complex and extensive. And one more thing is that the glycosylation profile is heterogeneous, whereas the proteins that we need, therapeutic proteins we need, should have homogeneous glycosylation profile. So in order to get that glycosylation profile, we need to have better understanding of post-translation modification. And we know from our literature that N-Glycosylation is one of the major post-translation modification, or we can say 50% of the post-translation modification are occurs in terms of N-Glycosylation. So understanding the N-Glycosylation pathway, what are the factors that affect the post-translation modification will help us to uh, modify the or produce a glycoprotein production platform that is uh, capable of using human cell lines for production of therapeutic proteins, glycoproteins. So within the N-glycosylation, this within the N-glycosylation, this video right now focus on. So let us understand what is where the N-glycosylation begins first. The N-glycosylation begins in endoplasmic reticulum, then it will move towards iric and then goes into the Golgi apparatus and then to the final destination path. And there are this integrate genes that will transfer the proteins from endoplasmic reticulum to iric to Golgi if the proteins have their native structure. If the proteins in during any part of this uh, secretion, that is from ER to iric to Golgi, if they have the non-native structures, then these retrograde proteins will transfer back the proteins into the endoplasmic reticulum where they can again acquire their native structure. And these are the different final destination of the proteins based on their structure. They are either diverted to the lysosomes or they diverted to the plasma membrane or secreted outside the cells. So within this post-translation modification, this video focuses on this part of endoplasmic reticulum, that is Calnexin and Calreticulum cycles. So this is the Calnexin and Calreticulum cycles. In this glycan structure is produced is already available in the ER lumen, part of which is formed outside the ER lumen and other part of it is formed within the ER lumen. So whenever the translation of the mRNA occurs, this OST helps binding this glycan to the specific site of specific sequence of the amino acid, that is NXST, where X can be any amino acid except proline. And this is now has three glucose attached to it. Glucose is represented here by a dark blue circle, and the glucosidase 1 and 2 help in trimming the this two glucose molecules, and this monoglucosylated glycoprotein can then be recognized by Calnexin and Calreticulum, and it will provide a isolated environment so that protein can acquire their native structure. Simultaneously, there are some ER57 or Cyp B, which is the member of PDI family or protein disulfide isomerase family, that helps the proteins to acquire their uh, native sulfide bond, which is essential for the proper functioning of the protein. If and after some time, this monoglucosylated proteins, the glucosidase 2 will remove the single glucose that is available on this glycan, so that now the proteins are no longer recognizable by Calnexin calreticulum, and they will move towards the UGGT1, where it acts as a sensor that can detect whether the protein has their acquired the native structure or not. If it has not acquired the native structure, then the glucosidase 2, if it is not acquired the native structure, then it will again add the glucose to the glycan structure so that it can again be recognized by the Calnexin calreticulum, and this cycle will continues. And after even after certain cycles, this when the protein does not get their native structure, the glucosidase 2 will trim off the mannose and uh, will trim off the mannose from this glycan structure, and mannose 6 to 9 is been recognized by when the structure has mannose around 6 to mannose 9, then it will be recognized and it is susceptible to be recognized by ERAD proteins that are that takes part in the ERAD mechanism, so that it can be now directed towards endoplasmic reticulum associated degradation. It will direct this towards proteasomal degradation. And if the protein acquires their native structure, then it will be directed towards ERIC as it is shown here, and for further post-translation modification. So this is the normal cycle of Calnexin and Calreticulin. So, however, because of some reason, if there is a increase in the amount of unfolded proteins, it might create a ER stress, and that ER stress will leads to epigenetic changes. So the slides now focuses on understanding the role of uh, chaperones that are involved in Calnexin calreticulum cycle. At the same time, this glucosidase 1 that is also involved in Calnexin and calreticulum cycle. So these three, that is MOGS or glucosidase 1, calreticulum, Calnexin roles has been studied in this, or will be discussed in this slide, in this presentation. Two approaches are being used to understand this role of this chaperones and glucosidase 1. First one is the proteomics approach. From this, the bioinformatic analysis has been done, and it has been found that there is a ER stress that is generated because of the absence of this chaperones or glucosidase 1. So this ER stress assay has been done to confirm that ER stress has been there or not. So first thing has been done is the proteomics analysis of different knockouts, and we found that the translation rate is decreased, and this translation rate decrease in translation rate is directly associated towards epigenetic changes. So what happens in Calnexin calreticulum cycle whenever the endoplasmic reticulum observe ER stress is that there is epigenetic changes, and cells will start to produce uh, proteins that are essential for cell survival at that point of time. That is, if there is unfolded increase in the amount of unfolded proteins, then it will try to increase the amount of proteins that are responsible in for ERAD mechanism, so that unfolded proteins will get reduced. At the same time, the proteins that are not involved in this cycles, uh, the translation of those proteins will be reduced. So this rectum translation, overall translation of the cells will get reduced, as it will stop translating proteins that are not required for cell survival at that point of time. And this in the top figure, it is shown that the translation of proteins uh, the translation of proteins towards membrane will also get reduced. So this indicates that the proteins that are not essential during this stress conditions are reduced. We can see that in calreticulum it is significant, whereas calnexin and MOGS it is not significant. So from this we can make an idea that ER stress could be high in calreticulum knockout, whereas it is low in calnexin and MOGS knockout. So how this ER stress conditions works is that in normal case, if the protein does not acquire their native structure, it will go towards this degradation. If protein acquires their native structure, it will go towards ERIC, and this is the protein translocation, that is signal recognition particle that will direct the proteins towards endoplasmic reticulum. So what happens in ER stress condition? If there is a increase in unfolded protein response, then the proteins that are responsible for this degradation will increase, and the proteins that are moved towards ERIC will decrease because there is a decrease in the translation rate of such proteins, and the proteins that are moved towards that are translocate towards endoplasmic reticulum will also reduced because of the ER stress condition, as it does not as at this point of time it needs to utilize energy uh, to get to come back to its normal state. So this figure represents the ER stress assay. So from the from this figure we think that at this point of time this ER stress is there in all the three knockouts, and ER stress is high in calreticulum, whereas it is low in calnexin and MOX compared to calreticulum. So first, this is the ER stress control assay. So what we have done is we use two different reporters, uh, one reporter, two different reporters that can gives the fluorescence signals whenever there is a ER stress. So first we check whether the reporters are working properly or not. So this is the control this first figure in A panel shows the signals of the cells when there is no reporter, and ER stress is artificially introduced with the help of thepsigargin. So we see that there is no signal. So in case of ER stress, cells does not have any signals, and it is confirmed by this in-QSAT fluorescence measurement and the fluorescence measurement with the help of flow cytometer. And in order to confirm that ER stress is there, we measure the CHOP proteins, that is the signature proteins that is produced during ER stress, and we found that ER stress is there, but signal is not available in the normal wild type cells in the absence of reporter. Then we introduce uh, one red fluorescence reporter, that is ATF4, and at different concentration of thepsigargin, and we found that the ER stress is there. However, ER stress for different concentration of thepsigargins is almost same, or we can say it's same, and this is confirmed with the help of fluorescence also. The ER stress in different concentration of thepsigargins are same. One of the reason could be that this thepsigargin affect different function of the ER, that is it affects the calcium homeostasis, and it is possible that in all the three concentrations the calcium homeostasis are being affected to a same extent as it is shown in the result, and maybe we confirm whether the cells has ER stress or not with the help of this western blot of this CHOP proteins are present or not, and we found that ER stress is there and signal is also there. So this ATF4 signal seems uh, is bona fide, and it represents the ER stress. It gives a signal whenever there is a ER stress. This is confirmed from this figure. Then we check the another reporter that is available in the literature that are available in the market, that is XBP1. This signal is also increased during ER stress. However, we found that the signals is quite low compared to the ATF4. as it is confirmed in both the NQ site measurements and also in the flow cytometer. And ER stress is there and almost at same level as the same concentration of thepsigargin are introduced, and the CHOP proteins are also present in these cells that are affected with XBP1. So since the signal of XBP1 is low compared to the signals of ATF4, we decided to proceed further with ATF4 for our actual experiment. And we did the experiment in this timeline. First, cells are infected with lentivirus that contain PLVX ATF4. Then they are incubated. The incubation is done for 2 days. Then puromycin selection cells. This plasmid contains contains antibiotic against puromycin. So the puromycin selection is done at 1 microgram per ml for 2 days. After that, they are transferred to the 24 well plate, and after 1 day, the experiments begin. The measurements of the uh, cells are being taken from both InQ site and flow cytometer. At the same time, samples are also collected for Western blot. So it has been found that calreticulum has highest ER stress compared to other Calnexin or MOGS knockout. And however, the right now this double knockouts ER stress has also been measured, but the bioinformatic analysis of this double knockout is yet to be done. So let us now not go through this Calnexin and Calreticulum double knockout. The Calreticulum has higher ER stress compared to MOX and Calnexin knockout, and this is in aligning with our bioinformatic analysis. And this data is also verified with the help of flow cytometer. The Calreticulum has higher ER stress compared to MOX or Calnexin knockout. And these are the control signals, that is cells that are transfected with ER stress, cells that are transfected with ATF4 plasmid, however does not have any ER stress, and these cells that are transfected with ATF4 and have ER stress. And this is also confirmed in the with the help of flow cytometer. And in order to ensure that these signals are not because of autofluorescence in the knockout, we also measured the cells ER stress. We also measured the cells ER stress when we also measured the cells fluorescence signals without when the ER stress is introduced in the knockouts that does not have fluorescence signals, in order to make sure that these signals are not because of autofluorescence in the knockouts. And we have found that there are no autofluorescence signals available. And in order to confirm whether this knockouts has ER stress or something else, we also measured the signature CHOP proteins that are usually produced whenever there is a ER stress. We have found that CHOP proteins is there in all the 4 knockouts: Calnexin, Calreticulum, MOX, and dual knockout. And this is the fluorescence signals from the NQ site, that is shown that Calreticulum has high ER stress compared to Calnexin. Um, these are the wild type that are transfected with ATF4, and thepsigargin has been introduced for ER stress. The next we did the glycomics study, that is we measure whether there is a change in the glycomics profile on the cell surface. For this we did a lectin study on the cell surface glycan, and measured them using flow cytometer. And we have found that around 57% of the statistically significant data are observed in the calreticulum knockout. as it is shown here. This Corne has been statistically significant in calreticulum knockout after Saridase treatment, and this RCA is not RCOA, this is RCA, this is typing mistake. This RCA data is also significant in calreticulum knockout, and in VVA the others knockouts are also significant, that is dual knockouts has more significance, but this calreticulum knockout is not significant. This significance data is compared uh, from the wild type signals. And this AAL, the calreticulum knockouts is significant in both solidase and non-solidase treatment, and this dual knockout is also significant. And one important thing that we have found out that in calreticulum knockout, slides are not properly arranged. In this case, sorry about that. Calreticulum knockout has more CD44 compared to the wild type. This shows that calreticulum knockout might have some presence or some resemblance with cancer cells, as CD44 is the marker for cancer cells. And so out of this all statistically significant data, 57% of the statistically significant are found in calreticulum knockout. We have in total observed 19 lectins and one this anti-CD44 proteins. So from this we have found out that there is a change in the glycomics profile in the cell surface, and also this is because of the disruption in the Calnexin and calreticulum cycles. So from this, and this experiment, that lectin experiment, is usually done on multi-lectin samples. So this data shows the overlapping of multi-lectin and single lectin samples to ensure that data are not uh, affected by data are not significantly affected by lectin interaction, or uh, yeah, data are not significantly affected by lectin interaction. So in the last two rows there are slight disturbance, uh, that could be because of lectin interaction. However, the trend is almost the same, that is both of them are increasing and both of them are high in both of them are high. So in terms of qualitative analysis, it does not give that much uh, disturbance. However, this data is not the data is does not show the ideal case. In ideal case, both of these signals should overlap each other, like in this case, like in this case. However, our data are still looks good enough that can be used for interpretation, as the sum of portion of these signals are overlapping. So what is the finding from these experiments or from this project is that calreticulum plays an important role in Calnexin and calreticulum cycle, as it creates more ER stress, as it is shown here, and around 57% of this statistical significant results are found in calreticulums from the surface glycan experiment. Calnexin ER stress is low. One of the reasons could be that calreticulum might be compensating the calnexin, and this MOGS knockout. In MOGS knockout also ER stress is low. In this case, the argument could be that glucosidase, since there is no glucosidase 1, there is no trimming of triglucosylated glycan structure occur, because glucosidase 1 starts the trimming of with the trimming of glucose first glucose that are present in the uh, glycan structure. So in that case, the protein's residence time within the Calnexin and calreticulum cycle decreases, and it will be directed towards ERAD mechanism. or E-red pathway in less duration of time compared to the wild type. And in double knockouts, one of the reason could be for less ER stress, is that the molecular sensor functioning might get affected because of the absence of both Calnexin and Calreticulin, as this UGGT1 and UGGT2 is also a glycoproteins. So this Calnexin and Calreticulin might play a role in stabilizing this UGGT1 and UGGT2 native structure. These things need to be validated from uh, proteomics analysis, or from proteomics bioinformatic analysis, or from some other experiments such as purifying these proteins from the knockouts and measure the glycomics profile. That could help us to identify whether there is a change in the native structure or change in their structure or not, or glycomics profile or not. As we know that the glycomics profile are usually changed whenever there is a change in native structure, so that different sites are exposed for glycosylation, as that is the same understanding, that is the same reason why we get different glycomics profile in the cell surface. So I hope you got some idea about this Calnexin and calreticulum cycles on this N-Glycosylation, and this information might be helpful in future for optimizing the therapeutic glycoprotein platform. That's all for this video. Thank you for your time.